on behalf of ICCB 2018, I have great pleasure in welcoming all of you, especially the panelists who really accepted our request at a very short notice. So I have great pleasure in welcoming Dr. Raghu Kalluri, Professor Ashok Venkatraman, Dr. Sundaram, and Dr. Pallavi Kshetrapal. So they are the ones who are physically present here. And then we have three distinguished panelists from who are joining from different parts of the country. Dr. Sudhir Gupta, he is from Mumbai. Dr. Sadhana Joshi from Pune, and Dr. Shinjini Bhatnagar from Delhi. Pallavi, you can come to and occupy the dais. Yeah. So I request Anurag to formally introduce and tell about the basis of this session.
Thank you very much. The basic idea of this session was very simple. Whenever we are talking to colleagues in the basic sciences, they point out the difficulty of connecting to clinicians. And sometimes I'm speaking to clinician friends, they talk of difficulty in connecting to basic scientists. So today we have before you a representation of the entire spectrum, basic scientists, clinicians, and people who do both. And I hope that part of the problem of getting people together is very obvious to you by seeing that three of our panelists, two of them clinicians, aren't here physically. And the solution is also obvious, use virtual media. So let me introduce, to start with, Professor Raghu Kalluri, Chairman and Professor of the Department of Cancer Biology and the Director of the Metastasis Research Center at the UT MD Anderson Cancer Center, where he also holds the Rebecca and Joseph Brown Endowed Chair, University of Texas. He is a Fellow of the American Society of Clinical Investigation and the American Association for Advancement of Science, awarded the Jacob Henny Medal 2015. Many of you may have heard of his seminal work in 2015 in Nature, looking at pancreatic cancer markers and exosomes. Professor Ashok Venkataraman is the Ursula Zulner Professor of Cancer Research and Director of the Medical Research Council Cancer Unit at University of Cambridge. He is a coordinator for the Center for Chemical Biology and Therapeutics at INSTEM Bangalore. So he is very well acquainted with both sides of the ocean and knowing how systems work in both places. He studies mechanisms of genome repair and replication and is an active translational researcher, translating these to therapeutic applications using novel chemical approaches. Professor Sudhir Gupta, who is on Skype, is a professor of medical oncology and deputy director at the Tata Memorial Hospital in Mumbai. He works on breast cancer, gynecological malignancies, and early drug development. As a member of the task force of ICMR, Indian Council of Medical Research, for guideline formulation on cervical cancer in India, he has headed and advises multiple clinical trials. Dr. Chala Sundaram, we are very pleased to have her here, is former head, Department of Pathology, Nizam's Institute of Medical Sciences. Her expertise lies in neuropathology, especially in diagnosis of neuro-oncology, neuroinfections, and neuromuscular diseases. She has been a very active collaborator with CCMB and has shown how these can be made. Professor Sadhana Joshi is the head, Department of Nutritional Medicine. That's Sudeep to the right, by the way. Interactive Research School for Health Affairs, Karsha, in Pune. Her studies have been invaluable in showing how maternal micronutrient balance will lead to altered epigenetic programming. You all heard of DOHAD. She's been a very important contributor, and that has implications for the entire life. Professor Shinjini Bhatnagar is right there in the little circle up there, and she's head of the Pediatric Biology Center and Dean of Clinical Research at Translational Health Science Technology Institute in Faridabad. She has made extensive contributions. For you, Shinjini, I don't have to read from this at all. Starting with work in rotavirus, and now she's heading molecular mechanisms of neonatal and maternal infections, growth, development, looking at preterm births, setting up big cohorts. We are so pleased to have you here. And Dr. Pallavi Kshetrapal, last but not the least, is a Ramalinga Swamy Fellow at THSTI. She studies pediatric health, and she also studies acute lymphocytic leukemia as a Ramalinga Swamy Fellow. She's been an instrumental part of cohort building and she brings a strong connection to CCMB, where she did her PhD. I'm so happy to have all of you here, and we'll get started.
So, quickly, I think I sent an email. So, we'll quickly start with this session where we want to showcase that how the interaction between a physician and basic scientist has helped in the field of cancer biology, and it has helped improve the patient care. So, we build upon that and then get on to the relatively unrecognized field of placental biology, but with huge potential, and see that what these few researchers and clinicians, they have their experiences to share, and how the experience of cancer biology can be utilized to use placenta and advance the research. So typically I'll request the panelists over here. You have 4 minutes to speak about your thoughts, and as I said, that I took the liberty of sharing our thoughts, that what you should probably be talking about, but you are free to say whatever you wish to or so. So 4 minutes for each of the speakers, and then we will throw the session open to the audience for question answer. Then we'll move on to the placental biology session, where the same way this will go, and then we will have a common discussion on this. So can I request Dr. Raghu to kindly come and share his thoughts?
Thank you very much. My name is Raghu Kumar. I'm from MD Anderson Cancer Center. I head the Department of Cancer Biology there. And I guess my task is to give you a feel of how basic science and clinical science or clinicians work together. Um, I'm an MD PhD, so obviously I've seen the two sides of the world. But I've been sort of lucky that when I was doing the medical training and when I did my science, it was quite obvious that the two worlds were a lot closer than maybe they would have been about 25 years before I did that. And I think that's a natural evolution of any um, aspect of life. And our interest here is science, that people tend to be in their own areas of interest and of their excellence. And as the knowledge evolves, we tend to throw barriers away and mingle. So I would say that in the world of cancer, that has been true, that clinical care was mainly disconnected with science. But I would say that in the last 25 years or so, that has come closer and closer very naturally, organically, because most of the clinical observations were tied to basic science discoveries, and basic science discoveries needed to be translated. Lately in cancer, I'm sure you heard of immunotherapy is a very popular treatment regimen, and it's really taken the world of cancer by by huge sort of a storm, in the sense of what impact it can have on patient care and outcome. Obviously, it's exciting because many cancers are responding to it, and there's a tremendous potential for even improving it. But if you look at the genesis of immunotherapy, it's a true example of how basic scientists did what they did and how ultimately it translated. If you talk to Jim Allison, he's a basic immunologist with biochemistry background, and all he wanted to learn was about T cells. And only in about the last 10 to 15 years is when he really began to think about translation. So my number one point I would like to make is that for any fundamental discovery in cancer, it is good to think about basic science as a very important endeavor. I say that because many people talk about this term called translation. And translation is a very important thing to do. That is to take something you observe on a basic science front and translate it to the clinic. But we cannot forget that all of us, be lost in the translation world and not think about fundamental discovery. So I think that right now, if I were to think about cancer science, I think that there's no more a distinction between basic science and clinical science. I think we should remove these phrases even, and we should talk about science as a continuum, which starts from an endeavor of wanting to unravel and find the mystery of something, and then that leads to identification of what you're going after, and then translating that into how it can impact the clinic, and then of course then the basic clinical work that translate all of this into practice in the sense of testing into patients. So that's the endeavor at MD Anderson. We don't really talk about basic science or clinical science. We talk about teams, and these teams are composed of everybody, including pathologists to surgeons to even nurses, because they actually deliver care and they're integral to even science that's going on. So I think that obviously, what else am I going to say? When you ask me, should basic sciences and clinical scientists should work? Yes, they should. But I think this is pretty natural now that the both sides should work. And you know, most of my basic scientist colleagues will tell you that fundamental discoveries in cancer were made by looking at Drosophila, looking at C. elegans, looking at all of these fundamental sort of developmental aspects there that led to major pathways in cancer that are deregulated. So what is very critical now is to think about the patient and think about new things we need to unravel and think about the teams you need to put into, and those teams can have all scientists with different expertise and clinicians who are also scientists in their own way to have that thinking. I want to end by mentioning that as much as basic scientists should think about where it's leading, because you cannot even publish a paper anymore these days in cancer, even as a basic scientist, unless you have a clinical correlation figure. Likewise, I think it's very important for clinicians to realize that this is a molecular medicine age. If, as a clinician, you're not prepared for your patient to come in with a zip drive and say, this is the entire sequence of my tumor that I got done for about $500 in China, can you tell me what's wrong? Which means that the physician must have enough knowledge to even understand what that is, give the right answer, guide the person instead of saying, I don't know what that is. So I think the clinicians also have to move towards, direction of the basic science. So I would say that I think that this is an exciting time for cancer. I think it's only exciting because everybody with an interest in cancer, whether basic science or clinicians, are working together. Thank you.
I think we got a fascinating view, particularly regarding building teams, centers, and learning from both sides. So, on that note, we move to Dr. Sudhir Gupta, who's a clinician researcher. And Sudhir, can you turn your video on?
Um, Actually, in the past about 10 or 15 years, there has been a significant amount of change that has happened in the practice of oncology. And I remember the time when I passed out and completed my DM and fellowship in 2000, the practice of oncology was largely empirical. The edifice of systemic therapy was essentially constructed on the base of chemotherapy and endocrine therapy. And chemotherapy, as we all know, is a very effective treatment. It has saved hundreds of thousands of lives, but it is not very targeted. It is not very focused, and it was largely empirical, including the choice of drugs as well as the regimens that were used. Now, what has happened in the past about 8 to 10 years is that we have had increasing amount of rationality come into the practice of oncology. And it started with a few cancers that were actually dependent on single pathways for their survival. And the classic examples are chronic myeloid leukemia and the PCR-ABL tyrosine kinase inhibition, the CD20 inhibition in B-cell lymphomas, and HER2 inhibition in a subgroup of patients with breast cancer. And those, of course, remain the poster stories of oncology in the past 15 years. What has also happened in the past about 5 years is that we have had increasing access to other types of targeted therapies. Most importantly, two classes of drugs. One is called the immune therapies, amongst them the most important class being immune checkpoint inhibitors, and then also a class of drugs that target the DNA repair deficient tumors. And all of this has essentially happened because there were significant biological insights into the nature of these tumors. Um, uh, some of you might know that the concept of targeting DNA repair deficiency started with the observation that some tumors cannot repair their DNA as efficiently as others can. And this, of course, is in some cases inherited in the germline. Hello, am I heard? Yeah. We have a class of drugs called the PARP inhibitors that have now, after a lapse of about 10 or 12 years, come into clinical practice in breast, ovarian, and a few other cancers. I want to submit before the audience a couple of points about significant challenges that we face in the integration of rational and biologically targeted treatment in the field of cancer. So, with few exceptions, most of these targeted therapies have actually been life prolonging but not curative. And the exceptions, of course, are CD20 inhibition, the HER2 inhibition, and PCR ABL tyrosine kinase inhibition in chronic myeloid leukemia. But most of the other treatments have actually extended lives of patients with metastatic cancer, but have yet to be proven to cure a fraction of patients with non-metastatic disease. And that actually is just a phase of development. We hope that in the next 4 or 5 years, we will have some clinical trials that will prove the curative potential of some of these drugs. The other challenge is that we have started on a quest of fragmenting earlier classes of tumors into biologically homogeneous subgroups. And what that has meant is that we have fewer patients whom we can recruit on classically designed clinical trials. So some of you would also know that oncology trials have in the past 20 years been of the sizes ranging from hundreds to thousands, and they have proven an improvement in endpoints such as disease-free or overall survival. But it is becoming increasingly difficult to conduct such trials. So, for example, if you have a subgroup of patients who have microsatellite instability high tumors, let's say amongst uterine cancers, how do you prove that such tumors will benefit from a particular treatment or not? You need, let's say, at least 600 patients to prove that point, but there aren't 600 patients that you can accrue on such trials. So I think with the advent of all these biological insights as well as treatments, we also need to evolve in the statistical planning of our trials, so that on the one side we don't have N of 1 trials, and on the other hand, we are also not encumbered with the need to accrue the number of patients that would be unfeasible to do. And the third most important challenge, just one more point, the third most important challenge is that there is a lack of access to some of these drugs because they are horrendously expensive. So, while they are significant advances, large fractions of population of cancer patients all over the world continue to lack access to these drugs. So I think those are some of the most prominent challenges that we will face going forward.
Need for high quality biology, high quality clinical, but the missing link in the middle is the actual therapeutics, right? Which brings us to chemicals. So, Ashok, what are your thoughts as a person who actually designs the type of things that go into treatment? What do you see as the link?
Thank you. I've already spent too much time talking to all of you. I've just delivered my lecture. So before I answer the question that was posed, I thought I would very succinctly try to cover some of the thoughts that I had that were not already spoken about by Raghu or Dr. Sandeep. So if I may, I'll start with that before I answer your question. So I hope that my talk illustrated what to me is simply inescapable. A connection between basic science and clinical science, not just in cancer biology, but across fields, is incredibly fertile and must be encouraged. It's happening all over the world. It must happen in India as well. I hope that many of the points I made in my talk illustrate the veracity of that thought. What is it that a basic biologist needs to do to try to ensure that this interface is cultivated? I have two thoughts. Many people ask wonderful questions which are really important, like how do cells move? Or how do cells divide? Perhaps it's also time to be asking questions like, why do patients who have this particular mutation develop this form of disease? In other words, look at the biology of disease rather than the biology of biological phenomena. And I think this is something that biologists perhaps could be encouraged to do, because there are many important problems out there relevant to disease biology. A second thought I wanted to share is, and I'm as guilty of this as anyone, I think, we believe that when we make a discovery, perhaps it's someone else's problem to translate that discovery to the clinic. Unfortunately, this doesn't happen. And very often, the onus is on the people who made the discovery to do what it takes, connect with clinicians, make the connections, move things towards translation. Even if you're not expert in the area, there are many people who are, but things won't move forward unless you pull people together. And I think Don Cleveland illustrated that very nicely in his talk. Conversely, on the side of clinicians, what are the sorts of things that can be done to encourage this interface? Almost as a mirror image of the first point I made regarding biologists asking questions about the biology of disease rather than questions about biological phenomena, I think it's the job of clinicians to emphasize, highlight unsolved problems in medicine that biologists can fruitfully tackle. Again, as a mirror image, I think clinicians, and Sandeep and Raghu both spoke to this point, need to engage with biologists to take possible solutions and advise and filter what is actually practical and possible and feasible in the clinic. So that brings me to the last point I want to make before answering your question, or attempting to answer your question, which is, from my limited experience of migrating perhaps between the UK and the India systems, what do I perceive as some of the challenges in India specifically? I think the first problem I'll allude to, I can term distance. I see in many of the premier research centers in India that the clinical research centers are not connected physically, with a few exceptions, to biological research centers, and vice versa. The most excellent biological research centers have no connections with hospitals or clinical practice. I don't think it's just a question of physical space. I also see a problem of what I call intellectual space. It's almost as if, to some degree, these communities are talking in slightly different languages and talking past each other rather than with each other. And I lay the blame actually at the door of Indian medical education, which I think the curriculum driven by the Indian Medical Council, and I think physicians will know exactly what I'm talking about, is so rigid that it stifles any attempt at originality during medical training, and it stifles attempts for young clinicians to get into research. But I think unless this happens, this intellectual bridge will not be crossed. The final point is infrastructure. And Sandeep alluded, for example, to issues like biorepositories, the need to develop sample collections, phenotyping on data. I think one thing that always strikes me is, how does the UK, with a relatively small population, have such a big impact on the genetics and genomics of human disease? And I think the answer is phenotyping. Because of the NHS, the extent of phenotype associated with genomic studies is huge and deep in a way that even much larger health systems like the United States at this point cannot replicate. And I think the lesson there is that in India, starting from these positions, deep phenotyping, clinically speaking, along with genomics, proteomics, and other things, is going to be vital. Funding, of course, is a major issue. Starting these endeavors is not easy. And I think what it requires is stable funding until these initial attempts have been given time to succeed or fail, followed by rigorous assessment to pick what avenues are most fruitful. Truncating, curtailing funding too early, I think, is not going to help to allow things to develop. So I'll end simply by attempting to answer your question. And I think the answer really comes from many of the things that I've said. The problems with small molecules particularly, and the challenge of developing new drugs, there aren't enough good targets. Now, you may be surprised. People will look at you and say, industry will look at you and say, but we have targets coming out of our ears. But I would retort, most of them are not what I would call good targets. In other words, druggable, rate-limiting steps in which you can expect a good therapeutic index and which are likely to yield a therapeutic phenotype. The second, of course, the second problem is the limitations of druggable space. And I've spoken about this before. There may be potentially tens of thousands of attractive targets for therapy encoded in the human proteome. We have the capacity to address with small molecules perhaps only a fraction of that space, and we need to understand how to do this. And finally, going back to the clinical issues, how do you phenotype? How do you decide how to stratify patients who are most likely to benefit from experimental therapies? For me, those are the three challenges. Thank you.
That's fantastically put. And Professor Chalasundaram, I invite you to share your views on how these connections can be made.
Good evening, everybody. As Dr. Gupta has said that in oncology practice is changing nowadays. From the cytotoxic, the chemoradiotherapy, now we have moved on to targeted molecular treatment and then to immunotherapy. So where do we fit as pathologists? We are a team player in this entire team where the scientific discoveries that are made by the basic scientists, we do on clinical samples to have the clinical application. So in this, the most challenging thing for us will be optimal utilization of the biopsy sample or whatever has to be collected, and how appropriately we collect and see that all these molecular tests can be done whichever. So the practice of pathology has changed a lot in the past few years. Just from giving a diagnosis to, these techniques and all have been incorporated for diagnosis, for prognostic markers, as well as for predictive markers. So the morphology may look the same. A glioblastoma, it may fulfill all the WHO criteria, but still the molecular subtyping is very different. So how best we take the sample and then see that the appropriate portions, that is, we have to avoid the necrotic areas, or whether we want the interface, or whether we are excluding the normal tissue, and what is the difference in the proteomics or the molecular features between a normal and interface and a tumor one. So that has to be seen under a microscope, and that is the area whether we subject it to a DNA or RNA extraction and other things. And in what instances we need to have a fresh tissue, and which tests we can do on a formalin fixed specimens, and how best to use these things. There are many tools that can be applied to the tissue, and at the same time we also have to know about the liquid biopsies. That is, at the time we may have to collect a whole blood, a plasma, serum, preserve it, these will be useful for monitoring the response to the treatment and other things. So all these have to be integrated, and when we validate any of these molecules by immunohistochemistry, it is also important for us to know which antibody to use and which clone has to be selected, what is the cutoff, how do we score those tests, and what is the result or the application that these people have. When we deal with a metastatic disease or a recurrent tumor, again, any resistance to treatment, whether which one we have to test for. And in a country like India, where the resources are limited, we also have to have an algorithmic approach. Otherwise, we cannot do so many tests on a limited sample, and it will be a cost effective also it should be. So we have to develop an algorithmic approach, which ones to do, which are cheaper, at the same time sensitive and specific. So we have to weigh the options amongst the various techniques that we apply, and these results will be interpreted in the context of a clinical scenario and not just giving a report. All the results, all the tests that we standardize, validate, apply, do, they have to be interpreted in the context. Because one gene, it can be abnormalities can be there in many other diseases, and so many may have the same phenotype. So that is how the crux of the problem is, and a pathologist has to be versatile both with the clinical applications as well as whatever is happening by the basic scientists who work with a controlled laboratory test. That environment is a little controlled, whereas in a clinical sample, it may be very, very diverse. So those have to be seen in this. Thank you.
Diverse and very interesting perspectives. I'll take the prerogative before I open it up to the audience of first agreeing wholeheartedly with Dr. Ashok that the medical education system is clearly partly to blame. And the languages being spoken by two groups are so different that even if you brought physical proximity, that would not in itself solve the problem. And I think what is required in these circumstances, perhaps, is the need of people who speak both languages. It would come from MD PhDs like Raghu. It would come from very informed clinicians like Sudhir. It could also come from those specialities which are trained on both sides. So, for example, involving a microbiologist or involving a pathologist, when a basic researcher plans a collaboration with a surgeon or a clinician, might be a better way of doing these things. So that's why I thought, your points particularly resonated, that pathologists clearly see both spectrum. And perhaps involving these specialties of medicine that somehow get overlooked in this basic versus clinical talk. We refer to them in India as paraclinical specialties. Perhaps engaging with them could provide one of the solutions. And I would love to hear your thoughts on that, Professor Lasundaram. Do you think that would be a role that could be done?
I think that is appropriate. All of us have to realize that we are in a team and everybody has to contribute. And not only this, and we also have to teach these basic methods and all, educate and bring awareness also. And I also bring out one thing that providing all these facilities in a reference lab also is not going to solve our issues. We may think that it is logical to have a reference laboratory to do some of the tests, but the pre-analytical issues cannot be addressed by a reference lab. So you may get a false negative result where the patient may be having a particular problem. Okay, so the fixation or whatever, everything to bring out that uniformity, we really have to involve in teaching and other things also before we validate these things.
So more than just phenotyping, all kinds of issues. So I think we can open it up to the audience and see whether they have any questions. Let's go there first. Let's get a mic.
See, apart from running a biology research lab, last 2 years I've been running an NGO on rare genetic disorders. And our major aim was to initiate, particularly interested in adult onset myopathies or other myopathies. And I will just relate our experience in this process. See, one thing we wanted to do was to actually figure it out how many patients are there about this. So we contacted a whole lot of neurologists. The response was very poor from most of them. We persuaded them. Finally, we got some data from actually contacting a very, very large, but the response has not been really all that great. We want to set up in fact, if you really look at it in terms of myopathies and others, there are hardly any center for neuromuscular diseases in India. So if you want to do any clinical research or lab-based research that goes into it, or if you eventually want to do clinical trials, you do need to set up centers of neuromuscular diseases in the country. I don't find any of them, except for one or two, they're very difficult to persuade neurologists to come together because they're so busy looking at a lot of other patients. I don't blame them. Stroke is, for example, one. So where, this thing. You will be surprised that even in All India Institute of Medical Science, you won't find a single clinician, neurologist who is interested in neuromuscular disorders. So overall, relentless working with people, and sometimes you feel very frustrated that you're not able to get anybody. Since I do basic science, I do understand the science behind it. So we are slowly going into this direction. In fact, the whole registry, mutation analysis, all this thing done by me and some of my colleagues. And I could not get any of the clinical geneticists, others to get involved. So what I'm basically saying is that somewhere we have to have an incentive system from our government. And in fact, I have even met a health minister regarding this, that have some special system by which you could, we could actually persuade some or other, persuade clinicians and others to get more involved.
So I think Dr. Alok is making the point that nature abhors a vacuum. Somebody will fill it. If the clinicians don't step forward, the basic scientists would do it. At the same time, do you see any problem with this approach? It's open. Let's start with perhaps Alok, because Ashok, you know the system well.
I go back to the point I made very briefly, phenotyping. So if you don't have deep phenotypes with some uniform parameters that can be extracted from health records, it's very difficult. And it's not just a problem in neuromuscular disorders, it's a problem in everything. The clinical sample, whatever analyses you do, if you don't know what the patient phenotype is, it's very difficult to work at any level. So I think that sort of thing needs, and I agree, national level effort. I don't think it can be done easily at lesser levels.
That you made when you were speaking, which you said that one of the solutions could be in talking in advance to the clinicians regarding what are the problems that bother you the most. And if the clinician says, look, right now in my clinic, it is not neuromuscular disease that is the real problem, it is XYZ that is the real problem, do you think that the basic researcher should then plan a career around that?
Yes and no. I don't think there should be any limit on basic curiosity-driven research. But there can be, at the level of government, incentives, particularly in a country like India, to encourage certain fields while remaining neutral about others.
I think for neuromuscular, I would like to say we had a very good collaboration with CCMB, especially with mitochondrial myopathies. Being a pathologist, we used to do the biopsies and we had good rapport with the clinical, that is the neurologist, and we used to give the sample. And for dystrophies and many other diseases, we had a very good rapport. And we could really have very good interaction as well as good research work out of this. Now in Nimhans, they have set up a very big neuromuscular lab where they are doing real good work on mitochondrial myopathies as well as dystrophies and other things. So it is how we interact with each other that matters more than whether it is a basic scientist, a neurologist, or a pathologist. I think that we should come together. That is the most important thing.
Can I? I think I'm going to strike a little bit of an optimistic note. And this is because I'd like to just brief everyone here about a new initiative that we're taking at the Indian Institute of Science, along with Christian Medical College, Vellore. Now, the support for this is to be able to bring clinicians and basic researchers together. And this is being funded with generous support from the Wellcome Trust DBT India Alliance in the form of a Margdarshi Fellowship that I'm holding currently. This took a lot of time to get off the ground. It involved multiple visits of I and my colleagues to CMC Vellore, where we met select clinicians who really were committed to answering questions in the areas that, or the problems that they face in their environment. One of the advantages of the Indian Institute of Science is that we don't just have biologists there. We have engineers, we have mathematicians, we have chemists. And our aim is to be able to get these clinicians talking to engineers, because engineers are very good at building devices, are very good at helping out clinicians in terms of making cost-friendly alternatives for the patients that they see in Vellore. Now, because of this fellowship, we've had common seminars where we've invited CMC Vellore clinicians to come. They spent as much as 2 days in the institute giving lectures. The engineering faculty, electrical engineers have formed interactions with them. Finally, coming out of this is this dire need in India for having clinician researchers. And therefore, in an MOU that we've signed with CMC Vellore, we are going to be strengthening an MD PhD program. Now, how do we propose to do this? We need to keep the Medical Council of India satisfied. So MD students registered in CMC Vellore for an MD will be enthused to come to IISc periodically, spend time in IISc. Should some of them wish to continue for a PhD, they will meet all MD requirements in CMC Vellore, and the degree will be awarded by CMC Vellore. They then will be transferred to the Indian Institute of Science, where their fellowship is going to be topped up so that it's commensurate with the seniority of these MD students. And we hope that they finish a PhD in 3 to 4 years in clinically driven projects. Not my favorite problem, but what the clinician needs.
I'll just ask Ashok only that you mentioned that you need to bring the clinician and the basic scientist in physical proximity. So I want to ask a little uncomfortable question to Sandhya, that being in Bangalore, why she had to go all the way to Vellore, because Bangalore is all full of hospitals. So what's your thought on that? And then maybe Ashok can talk about it. Yeah.
This was the question that I was asked during the presentation to the Wellcome Trust DBT India Alliance. They refused to accept that Vellore was going to be proximally close enough to Bangalore to get stuff done. It's not any clinician. It's not any hospital. You need to find the right type of clinician who's interested in believing that research will contribute to their clinical practice.
Yeah, I think that stresses the point that Ashok
exactly.
made.
And that is what we found in CMC Vellore.
It's not physical distance alone, but intellectual distance as well. And I have only one very practical comment on what Sandhya said, which is terrific. As someone who trained at CMC in medicine, I know that actually the postgraduate trainees drive the clinical service. And the big problem is that when you take people out of that system and get them into PhDs and laboratory-based work, their colleagues complain because the clinical load goes up. So I think funding replacement of clinical services to encourage this kind of thing is something which is really vital. That was the practical point I wanted to make.
Okay.
It's a service.
Actually,
Good evening.
Could I add a small comment? Just a quick comment about the interactions in Bangalore. I'm from Bangalore, from the National Center for Biological Sciences and INSTEM, and we have a major collaboration with the National Institutes of Mental Health and the neurosciences across 5 different clinics. And I echo all the concerns raised, but I think basic scientists and clinicians have to go at at least halfway, if not, and more, in order to engage. And whether you're in the same city, and as Sandhya says, whether you're far away, it's actually that walking that distance, but also engaging with the clinical people who are interested in asking questions, and the basic scientists, not simply being
point taken, Jitu. I mean, this is the point that we were trying to drive, that just physical proximity alone wouldn't really
alone doesn't work.
So
but
we are really, going, I mean, it's a very interesting session. We'll just take this last question, then we will go to the placenta.
Good evening. I am Dr. Prasad. I thank both the gentlemen who came on either side of the Atlantic Ocean countries. One is from USA and another from UK. Since few months, rather to say few years, I am thinking of lot of health-related subjects in India. Particularly the 75% population is living in the rural and tribal areas. That is totally neglected. They are facing a lot of problems. Majority of the corporate hospitals are in the cities. The persons who cannot afford the corporate hospitals are still suffering. The air we are breathing, the water we are drinking, the foods we are eating is totally contaminated. We are growing the foods.
Excuse me, sir.
Full of.
May I request to just stick to the session, because
wait, sir. Please give me some time, sir. Okay, let me speak. Okay, sir. Then we are not having the cancers. All kinds of diseases we are having and we are suffering. As the subject of the cancer is more here today, we are supposed to look for the alternative avenues, prevention, other measures, immunity medicines which are enhancing their immunity such that the entire process is coming down. Apart from that, I suggest another thing is also alternative medicines. We are supposed to look into the matter. Some of the herbal things, herbal medicinal things are also in usage in this country, where further research is essential. I am requesting the CCME to take care of this kind of research work, what is going to be helpful, where a chemotherapy effect is going to be reduced, ill effect. That is my suggestion, because as he is telling, a lot of time is the factor here. That's why I am supposed to cut down. October 8th last year, I conducted a meeting in Rangaraya Medical College of Kakinada, where I have invited Dr. Radha from CCMB, and I think Kolluri Subbrao, biochemistry professor, I think your father, Mr. Ravi, he is from, your father also came to my meeting, the former vice chancellor of NTR University, all the medical college professors, we had gathered, we formed the academic consortium for medical research, and we are doing this one. I request, medical colleges are not doing any research work. I want CCMB should come forward to help them.
Rakesh, I think you will agree that CCMB will take up this role. Okay. Thank you, Dr. Prasad. We will really take up your request very seriously. So, thanks everybody for participating in this session. And no, you can be there, please. Please be there. It's all right. I mean, we still want your wise words on placental biology thing. So, as I said at the beginning, that want to take the, how the success has led in the patient care in cancer, we want to just understand a bit on placental biology. So I'll request Dr. Sadhana Joshi to share her experience. So she is a basic scientist and she works very closely with clinicians. So let us hear what her experience has been in field of placental biology.
Yeah, actually, we are working on pregnancy complications. One of them being preeclampsia, which is characterized by a placental pathology, the high blood pressure and proteinuria. So I have not heard earlier, but I think you all know that the placenta carries marks of events that have occurred during pregnancy. It is not just a transporting organ, but it is possibly programmed to optimize the pregnancy outcome. So if we understand the placental growth and development in itself will show us the fetal status of maybe nutrient sufficiency or insufficiency inside the womb. What we have done is we have shown in preeclampsia that it is the nutrient imbalance, that is folate, vitamin B12, and maybe omega-3 fatty acids. They can create epigenetic marks on the placenta. So we have looked into angiogenic factors like vascular endothelial growth factor, and shown that this has been methylated in the placenta. This has got implications for the long-term health of the fetus because we all know that children who are born to mothers with preeclampsia or they are born preterm, they are at increased risk of developing neurodevelopmental disorders. So what we could possibly do is, like in the case of autism, studies, a very recent study has shown that the methylation in the placenta can predict for autism. So if we can try and expand this to larger horizon, like maybe cardiovascular diseases, because we have already shown that in preeclampsia, in the placenta, these angiogenic factors are getting methylated, and they can be modulated through the diet.
Sadhna.
This gives us some scope of trying to intervene in some point and change the methylation marks on the placenta. So these.
Sadhna. So
yeah.
Thanks for sharing your huge experience in this. We were just also interested to know, because you have been placed in the medical college and hospital, and it was
Right.
quite early on that about 10-15 years back that you started interacting with the clinician. Can you share your experience that how difficult it was to convince the clinicians, and how could you really reach this far?
Yeah, actually, see, clinicians are busy with their OPDs, IPDs. So initially, it was very difficult trying to convince them that what are you going to do out of the placenta, which is, kind of a throwaway organ, which has no meaning for them at that point of time. And trying to understand that they need to give us the placenta. The other thing is the placenta, anything that we have meaningful out of the placenta, it has to be processed very quickly. So at the time of delivery, the constraint that they have is that they are more have to look after the mother as well as the baby. So in those circumstances, for them trying to look after the placenta and make sure there's somebody, you call the clinic, the scientist, and then you are present over there. So it creates a huge amount of difficulty. In fact, we still are facing a lot of problems because they have their own constraints, and the time period when the placenta has to be processed is also very small. So, but I think if we have edge now, the advantage that we had is we were based in the medical college.
So
So it
Sadhna,
was a little.
actually, you had to enter the operation theater, enter into the
Yes,
domain of the clinicians to really get the material out. So quickly,
yes.
do you think that things have changed now, and the clinicians really think that they come forward to you for asking any relevant questions in this field?
Yes, I think things have changed. A lot of clinicians are now coming up and they have made a lot of effort, I think, to understand what we are trying to say. Initially, this concept of programming itself was a little too difficult. It's not in the classical textbooks of medicine. But now I think they're coming around and they themselves also raise a couple of questions. So I think we are progressing.
Okay, thanks a lot, Sadhna. Please be online. We will get back to you again. Yeah. Okay.
Okay. Thank you.
So clearly, I mean, about 3 decades back when I graduated as medical graduate, placenta was considered as a waste paper basket tissue, and we would just allow it to throw straight into the dustbin. Things have really changed remarkably from there. And Dr. Shinjini Bhatnagar at THSTI, she has been one of the pioneers in this field. So I request Shinjini and followed by Pallavi, because they both work as a team. I request Shinjini to start, and then maybe Pallavi can take over from there. Shinjini, please.
Okay. We had put together some slides, and so I'm just going to. No, I'm going to skip. I'm just going to go to the third one.
Okay. Evolution of
And
placenta.
yeah, so I think Sadhna's covered I think we all know. It's interesting as I'm a pediatrician by training, and I'm now involved with a lot of child health research, have been for the last 3 decades. But it's interesting how through my pediatric training as well as early years of my research, placenta was always looked at as an organ which was really the end of a disease. And it's been, but if you look at it, it's one of the most crucial things. It's interesting. It's a transient organ, and yet it's the cornerstone of pregnancy, the fetus, and the subsequent growth and birth, and the growth of the child. And despite that, very little is known about this organ. So it's very heartening to see that now there's a lot of interest and we're moving forward in this area. So basically, I'm going to let Pallavi handle what she has taken up this program in a big way. I just want to tell you that you asked this question from Sadhna, how was it to get into a OPD or a labor room? And so what we've done is that we've created a platform where the clinicians and the biologists can work together in being able to take this whole question of how important is the placenta. And talking about the placenta from a dustbin tissue to possible treasure trove, is it possible? And there are two areas that we've been working on. One is the omics approach, and the other is to develop safe, effective ways to assess placental health in real time. We must note that the placenta comes when the pregnancy is over. So we really don't know when changes have happened to affect the fetus, and therefore real-time images of the ultrasound is to capture the morphology and the functions in terms of blood flow is a very important way forward. So we've set up the Pediatric Biology Center at the Translational Health Science Technology Institute at the NCR Biotech Bioscience Cluster, which is an interdisciplinary center which is dedicated to maternal and child health and has clinicians and biologists working towards childhood health and disease. And we've created links. We don't have a hospital of our own, but we've created links with the tertiary hospitals as seen in the slide. Are you on that slide?
Yeah.
The one, yeah.
Yeah, that's right. Yeah.
A more important link that we have is with the district hospital at Gurugram, the Gurugram Civil Hospital. So we are actually capturing the population as well as the participants who are getting referred to tertiary hospital. The next slide, if I can have that, please.
Yeah, it's there.
Yeah. So we've set up, this is as part of a national initiative, we've set up a multi-institutional interdisciplinary program on preterm birth. Our objectives, of course, are larger: to do an appropriate risk classification of women for preterm birth, looking at clinical, environmental, and biological factors, and do an accurate categorization of the preterm birth phenotype, and then develop an effective algorithm for diagnosis and timing of interventions for preterm birth. But as a secondary objective in this whole cohort, we have now created a platform to study the placenta. And this program has Safdarjung Hospital, Maulana Azad Medical College, and from the research institutes, National Institute of Biomedical Genomics, Regional Center for Biotechnology, and is a beautiful mix of clinical epidemiologists, statisticians, geneticists, proteomic and imaging scientists. And from this, using this platform, we've established a pregnancy cohort. So when Sadhna was talking about the research, a lot of research that's been done on the placenta is cross-sectional. And today what we need to understand is in a series, in a longitudinal series, what is happening ideally from preconception, but here from the first trimester and as we move forward towards childbirth. And so we've set up this cohort where we enroll women early in pregnancy, follow them till delivery and postpartum period 6 months after that. And we're collecting, yeah.
Okay, so quickly, I mean, you said that you have been now able to set up this center. So because you had an advantage of being a clinician, do you think a basic biologist, if he had imagined about, this sort of a work, what hurdles or what challenges that individual would have faced?
I think I'll leave Pallavi to answer that, because Pallavi will tell you how, when she came into our picture, what happened. All I want to say is that, unless for us, it's not been a clinician coming to a biologist to get their samples processed, or a biologist coming to a clinician to ask for samples. It has, and I think that's the bottom line. It has to be a question which has to be thought through together and then taken through together. And I don't think we face problems because it's been a team right from the beginning. And I think it'll be, if once Pallavi tells you her story, it'll be clearer as to how we've set up this whole program.
Thanks, Shinjini. And so we clearly heard that how Shinjini came out with some solution about trying to get into the placental biology field, taking care of the lacunae which actually exist in this field. And I think that sets the stage for probably the youngest panelist on this session to share her experience as basic scientist as well as the young investigator, as I mentioned. Yeah.
So this was the topic given to me here. It's about how are the opportunities and challenges in the field of placental biology, especially with reference to translational aspects. And if you think that I have put in a wrong figure here, that's right, because I am a developmental biologist trained in Drosophila, and I wish Drosophila had a uterus, I would not have to move. So this was, you can say, a marriage made, it's like an arranged marriage that I really wanted to work in child and maternal health. So what I did was I found the right type of platform, and that platform, what Shinjini meant was THSTI, where we are built up with different centers. So we have clinicians, we have immunobiologists, we have biologists, we have pathologists with the clinicians, and we also have Center for Biodesign, which make low cost effective devices. So that platform was already set. So now when you go to the clinicians, they say, we don't have time, and why do we give you the samples? So if you become the sample taker, in the sense you yourself put into it that, okay, we will discuss about the strategy, how the samples have to be collected, make your own SOPs, go to them which are feasible and easy for them to understand, and also make them understand the question that why this sample has to be collected, as you said, this way or that way. So make your SOPs which are very tight, be transparent with them, and I think that works. So if I can get my next slide, I'll be very quick. So to understand placenta, the first thing was, I was in a platform where I got it, but I needed to know more about it, and that was nationally and internationally. So what we did was, in 2015, we had this meeting, which was an invite-only meeting, where we called all the people who were interested or vaguely interested in working on the placenta, but had also hands-on with new technologies. Like Shinjini's mentioned, that placenta is a dead box. It's like a black box. The thing has already happened. How do you get the information from it? So can you go a little back to trace the history? And to trace the history, we wanted to know what are the new technologies which people are using for real time and to understand the organ. So we called people from NICHD, NIH, and from India we had Sadhna Joshi, from Bangalore we had CMC Vellore coming. So there were all these different locations which and Pune, Dr. Yagnik had come. So then we thought about this old problem, which is the placenta problem, to understand adverse pregnancy outcomes and new approaches. So that was a new marriage we were trying to think about. And that was new technologies for the real-time assessment, what Shinjini has already pointed out, and identify biomarkers for placental function in maternal biopsy. So basically tracing back the events which have happened. So challenges we have not faced, as Shinjini said, because we were...
So Pallavi, I have a provocative question for you. You were very fortunate, that you were born 20 years later than I was born. Suppose if you didn't have this platform, what would you have done? Would you have gone back to Drosophila, or you were really excited about this question so that, you moved to this field?
So the Drosophila had to lose its wings. So what I did was, when I was doing my postdoc, I translated a little bit over there to work on the metastatic tissues. So when I came back, this platform wasn't there. I am a Ramalinga Swami fellow. So when I came into this place and this was going to be built, I also went to clinicians at AIIMS to work on pediatric B-cell acute lymphoblastic leukemias. So there is where I found...
So it's not only your experience. I'm saying that, say, if such a person comes, doesn't find this field, what do you think would be the challenge?
So the challenges are there, as everybody has mentioned over here, but I like the optimism which Sandhya Vishweshwaraya ma'am had presented here, that curiosity-driven research, bring the right type of people together, team up. So if you don't have geographically located areas, go ahead, team up with the right type of people who understand you, and multidisciplinary, and it's up to the biologists. So what I do is, clinicians are very busy with the national load. So it's up to the biologist here. I take that thing. to help translate the novel concepts of molecular biology, which we talk about, into applications that could be used in the clinics.
So what do you think are the challenges? What is the other opportunities that this placental biology field provides?
Yeah, so if you go 2 steps further, I think I just have one slide which tells you about how placenta can be used for this. So yeah, so this was the dissections which
Maybe
we
you
were doing.
speak of.
Yeah, the last, what the point, yeah, this one, no, just back, yeah. So you can analyze the placental function. So what we have in our lab now is a candidate gene approach, the red one, to understand at least at one time one gene till we have the cohort ready. So that is already done. Now we are at the green stage, which is the association studies where the placental shape, measurement by ultrasonography, and the flow, that is the pulsatory index, which could tell you the placental function. Now correlate that placental functions, as ma'am pointed out, by algorithms. You can't have one gene doing everything. So it has to be a panel. Everybody is going ahead with a panel. So that panel could be with the help of the clinical parameters and the biological parameters. Can we come up with algorithms from that?
Okay. Thank you, Pallavi.
Welcome.
Now we throw the session open for questions if anybody has.
Okay, great. I just wanted to make a couple comments. So I used to be at NICHD.
And I heard your talk.
Great. Actually, I got enticed into doing some placental research while I was at NICHD.
And I heard your talk at NIH also.
Oh, you did? Okay, great. So I want to, there's a lot of aspects of the placental system that are really fascinating cell biologically. And the only reason why we sort of got interested in it was through a mechanism that I want to just throw out for you to consider in your various departments. And that is, if you really want to have sort of creative input into a field like placental biology, one thing that you might do is have a creative incentive mechanism for giving grants for research in that area. It was really through that mechanism at NIH that I came into that field from a cell biological perspective, because we realized, hey, this is a really neat system because the placenta is a syncytium. And, what are the fusogens that are involved? What is the process of syncytial formation? And so we were able to get those trophoblasts and watch them fuse, just sort of go at a very basic cell biological level. Now, to what extent that impacts these global issues in terms of how important, how you deal with the health defects is, a long way away. But I think it's an interesting mechanism, these sort of incentive mechanisms for putting out a call and say, look, we're going to fund a lab who may not have any placental background, but we will fund you, to the tune of a postdoc or, a couple of graduate students, and you will find that there's probably people who are willing to think about placenta in order to sort of to take on that opportunity. And it has worked very well at NIH, not only for placenta, but for HIV research.
So you have 2 questions. I'll answer the first one about the funding for the placenta. So this meeting was where we also invited the DBT secretary, the DST, the ICMR. So these are the funding bodies at India. And thankfully, an RFA was called after the meeting because this was published. So they understood the impact of it. And there were 2 grants which were funded, one which I got, and the other one is from SGPGI. As you mentioned, they didn't know about the placenta, but the technologies were where they were isolating exosomes from the urine for diabetes. But now the field is such that because they can collaborate with this, because it's a hospital, so they collaborate with the gynecologist, and now they are going to do the same thing with the GDM. That is the gestational diabetes mellitus, if that is affecting the baby or the outcome by a preterm birth, because that was mostly in terms with preterm here when this call was called. So that was the second question. Hope I've answered the 2 of them.
Okay, Ashok. pass on the mic.
I'll be brief. I just wanted to say that this conversation has perhaps illustrated some of the points that were discussed in the first session. So I think Shinjani and Pallavi have really illustrated how it is possible to build a center that is multidisciplinary and brings multidisciplinary basic sciences to ask or solve important questions concerning, in this case, maternal and child health in India. And I think Jennifer's point raises another issue. How does one incentivize? Resource is not infinite, especially in a country like India. So I think choosing some of the problems that are clearly relevant to the health of India and perhaps encouraging, stimulating basic research, incentivize was the word that Jennifer used, I think is a great idea.
Yeah, I would just add to that, Ashok, that you're very correct that, over a period of time, the maternal health and its influence on the child, particularly the effect of maternal nutrition on child health and future consequences, that is really come to be recognized because of some work that has been done in this country. And that's why I think there is a great recognition about this placenta. And I just like to mention 2 points: that placenta allows to understand the direct effect on the maternal health, as well as it acts as a physiological barrier. I mean, we think it's physiological barrier, but it can actually be working so pathologically that it can transmit all these risks to the child as well. So thanks for making this point again also. So is there any other question?
Yeah, I'll just comment on Ashok's question about incentivizing. So just to let you know that this cohort itself is a national cohort and it's been funded through the Department of Biotechnology, Ministry of Science and Technology. So we augmented this because we never thought about, we knew that we are going to collect specimens. We were collecting 19 different types of specimens at 5 different time points of the pregnancy as postpartum. So now we have 5 lakh samples, different aliquots, which are maintained in 30 deep freezers. And those 30 deep freezers are monitored by remote temperature monitoring. So sitting right here, we can get to know that what is it. So that becomes a very big national, as you said, that you have to really you have to sort of incentivize and wait for the platform to be built before you can get things churning out of it.
Okay, Pallavi, I'm sorry. We will have to. I think this sets a great stage for, more discussion over something more exciting, which is just behind you. I'll request Anurag to just summarize this session and maybe finish there.
Yeah, I think the points are obvious. Pick a good question for which you can make a compelling story against competing projects. Pick great partners. Be willing to meet them halfway. Flesh out your vision. Don't just imagine 2 of you can do it alone. Try to find intermediaries if you don't speak the same language. Identify the funding, bring in the funders. Do it well and do it properly. That, I think, to my mind, was the biggest summary. But sit and plan and talk and discuss. Physical proximity is important. Mental proximity is even more important. Find them in Vellore if you have to. Find them in another country if you have to, but you have to do this. Thank you all.